Can everybody hear me in the back? If you just walked in the door, wave your hand so I know that you can hear me. Oh, he doesn't follow direction well. Well, welcome to Down to a Science. Tonight we're talking about memory and aging, or I like to call it, forget about it. So welcome. We have a packed house tonight. I'm excited for the talk tonight. Who's ever been to a science cafe before? Wow, half the audience. That's about as good as it's ever been. So for those of you who have never been to a science cafe before, it's about the scientists coming onto your turf, answering your questions, and we're going to have a discussion about what it's like, what happens to our memory as we age normally. So it should be an exciting conversation tonight. You may not remember it at all tomorrow, but it's good for the rest of the night. A couple quick logistical points for you. This is my friend Jeff Miller. He's going to actually do a short interview with our speaker tonight, Dr. Adam Gazzaley, to start off the evening. And then we're going to take a few questions from the audience, and then Adam's going to give a short presentation about his research into memory and aging. And then from there on, it's discussion time. Your questions, your experiences, your chance to actually maybe get a little consultation from the neurologist up here. So it should be a lot of fun. About halfway through, we'll take a break. Be sure to order lots of food and get lots of drink from our gracious host, the Atlas Cafe, tonight. Big hand for them. Always do a great job for us here. Last couple notes. I have a mailing list that will keep you up to date on everything that's happening at my Science Cafe, all of our monthly events, all the tours that we do. This man up here in the front is waving her around. I'm going to have him pass it around. Sean, can you pass that around? Oh, look at that. You follow direction well. And just name, email, and there's also an entry form if you want to be on a weekly email newsletter about science events that are happening in the Bay Area. One quick plug. November 1st and 2nd is Wonderfest, the Bay Area Festival of Science. November 1st is at Stanford. November 2nd is at Cal Berkeley. There are some amazing, amazing speakers. I think only 3 Nobel laureates will be speaking that weekend. Only 3. So there's some great conversations from energy policy to cancer research and everything in between. I hope you check it out, wonderfest.org. Do you have any questions for me before we get going? Excellent. Dead silence. So I want to introduce Jeff Miller, who is now head of public affairs for the Lawrence Berkeley Laboratory.
Formerly at UCSF, now head of public affairs at Berkeley Lab. So I'm going to do a quick focus group. Raise your hands if you have heard of Berkeley Lab before. Hey. All right. Very good. Yeah, I haven't done anything. I've only been in 6 weeks. So I'm going to be your stand-in here for about 15 minutes doing this interview with Adam, who is, as you will see, the real brains of the operation. And then there will be some time to ask questions. And since my brain is failing as I age, I'm actually going to work from notes tonight. So at the end of a long day, I figured that was okay, and you wouldn't mind. So if any of you have listened to the Science Cafe podcast, which I did with Adam last March, some of this might be a little repetitive, but I'm going to assume that most of you have not. So, Adam, good to see you again. So why study the brain and not the heart or immunology or any of the other many things that I'm sure you were interested along your MD, PhD career?
Well, that's a good question. I started with a very... Can everyone hear me? No. Hello? Oh, wait.
You can use this for a second.
Hello? Hello? Okay. So I started with a very general interest in science when I was growing up, and I was actually interested in astronomy all throughout college, and switched my focus to an interest in the brain, largely because it had all the complexity that I felt looking at astronomy and thinking about space had, but our brains are responsible for every aspect of our behavior, everything we do when we move, we feel, we think, all our emotions. So just in terms of its complexity and also the personal aspect of it, in that we are really our brains, is what attracted me to it.
So why not the developing brain instead of the aging brain? Also a very good and sexy topic these days.
Yeah, I actually fell into aging research by chance. My advisor, when I started grad school in, I guess, '92, just had recently started being interested in aging. And when he first approached me with the idea of doing my thesis work on aging, I was not interested. I was 22 years old and felt like I'd never get older. So first I wasn't, I was like, aging doesn't really apply to me, although that's an idea that's clearly changing now, and younger people are thinking about aging a lot more. But after reading about it for just a short while, I got really interested in it and realized that it is the future in so many ways. So I decided it would be the thing.
So there was no single defining moment in case any current or prospective parents want to encourage their offspring to become neuroscientists. There was no chemistry set or...
Yeah, I was actually a super nerdy kid and had a chemistry set and a ham radio and a physics set and a biology set and all those things, and got in a lot of trouble doing experiments at home.
Okay, well, speaking of experiments, this is going to be a pitch for money, actually not, but the technology that you use to study the brain, how has that changed in the last 10 years or so? And how expensive is it really? Because we hear a lot about the costs of research.
Yeah, it's fairly expensive, especially some of the tools that we use. I'm going to show you some slides about sort of the cutting edge of neuroscience, what we use in our lab, but we use a functional MRI machine, and it's a 3 Tesla magnet. It's actually responsible for setting one up here and actually purchasing one for UCSF, and that's around $3 million. Tried to put that on my credit card and get all that frequent flyer mileage.
See, we're going to be passing the hat here before the evening's over. So why is it that evolution has decreed that our brains must dim? My 92-year-old father always complains about his brain can't turn over like the engine in the car he shouldn't be driving, but so why?
Well, I feel that part of that is that our brains, at least from the human perspective, our brains never really had the evolutionary driving force to survive for this long, right? People, even at the turn of the century, when we were living till a little over 40 years of age. So we don't really have the mechanisms to keep the brain perfectly healthy throughout an entire lifespan. But it can't be the only reason, because if you look at animals that don't live very long, like rats and mice, and even monkeys that live into their mid-20s, all animals, not just humans, suffer a decrease in cognitive ability. So it seems to be a general feature of aging, and that systems, all organ systems, degenerate to some degree, and the brain is not immune from that.
Well, one of the questions that I hear a lot, and I know you must be really tired of hearing, and I can anticipate some would like to ask. So you become more forgetful over time, and let's just say that there might be some brain disease in your family. How can you actually determine, or can you, when it's just normal aging or something more serious like dementia or Alzheimer's?
Yeah, that is the ultimate question. So I still do see patients at UCSF, not so frequently since I run a lab, and that's most of my time. And I see exactly those people. These are older individuals that are worried that their thinking and their memory attention is just not what it used to be. And they're like, is this normal, or is this something to really be concerned about? And there's no great answer for that question generally. And that's why we see these patients, and that's why we do the research we do. So trying to dissociate what's normal from the very earliest stages of things like Alzheimer's disease and other dementias is a real challenge, and we don't have any answer for that right now.
So for purposes of clarity, how would you define Alzheimer's and then dementia?
So this is a question that I'm asked a lot, and I think it's frequently confused. I think you could think of aging as having these different levels upon which cognition changes. So one that we study in our lab we'll define as normal aging, and that's what happens to most people as they get older. Actually, normal aging is really the aging processes that occur that if they didn't would be abnormal. So for instance, as women get older, they go through menopause. If they didn't go through menopause, it would actually be abnormal. What we tend to think of as normal aging is usual aging. It could have disease associated with it, like coronary artery disease. Some degree of plaque buildup is almost common throughout, almost universal throughout the population. So you can think of normal and usual aging. Then you have the other side of the spectrum, and you have dementia and Alzheimer's disease. It's important to realize that dementia is not a disease. Dementia is a syndrome. And a syndrome is a collection of signs and symptoms that could be caused by a disease. So in the case of dementia, the syndrome is a decrease in your cognitive abilities. So dementia is not a developmental disease, it's a degenerative disease. So you have to, a degenerative syndrome. So you have to start at some level and then decline. And it's really defined that you've declined to the level that you're now impaired, either occupationally or socially. And that's a really hard thing to ascertain sometimes, because depending on what you're doing in your daily life, it might be hard to tell if you're functionally impaired. So if you're retired, let's say you're sitting on the couch and watching TV, it could be a long time until you're functionally impaired from that, right? But if you're still working and engaging in very rigorous activities and competing on the workforce with 20-year-olds, then it might be a lot earlier. So that's dementia. It's still not a great way of defining it, but that's how it's defined. And like I said, many diseases cause dementia, and the most common of all is Alzheimer's disease. So Alzheimer's is incredibly common. Once you're over 50 years of age, once you're over 80 years of age, it's almost 50% of the population. So it's very common. A lot of times people have Alzheimer's disease and we don't know it, because Alzheimer's is defined as someone that has dementia and then the other signs of Alzheimer's disease. So there's no real way of definitively diagnosing Alzheimer's disease without looking at the brain itself. So a lot of times, and this is what leads to the complications, is that people are concerned they may have Alzheimer's disease, but we don't know it yet. So you could think of those sort of aspects of what aging and the brain is about.
So would you care to speculate on what current theories about what might cause Alzheimer's?
Yeah, so we know the pathological changes of Alzheimer's disease, things like amyloid plaque and neurofibrillary tangles. And essentially these are protein accumulations that are abnormal in the brain that are associated with cell death. Why these proteins accumulate, we're not sure yet. There are certainly some forms of Alzheimer's disease that are genetic and what we call familial. This is by far the vast minority of Alzheimer's disease. These individuals usually have Alzheimer's much earlier, in the 50s. It's usually a little more rapidly progressive than standard Alzheimer's. But most Alzheimer's occurs in people older than 65 usually, and is not genetic. So another, just because it popped in my head, another thing that I'm frequently asked is people are worried because their mother has Alzheimer's disease or their father, and then they're like, should I be worried that I'm going to get Alzheimer's disease? Well, if your parent got Alzheimer's disease in the older years, like let's say 70 years old, you're really at no increased risk, because with Alzheimer's being as common as it is, that doesn't put you in any increased population. Some scientists think that if everyone lived long enough, then everyone would get Alzheimer's disease eventually.
Okay, now we're going to do a little true-false test with Adam. So are you ready? The brain shrinks as it ages. True or false?
True. The brain does shrink, and it doesn't just shrink in individuals that are older, let's say older than 60. The brain really starts shrinking in the 20s on. So it's pretty much a linear process throughout life where the actual size of the brain declines.
It's not just drugs and alcohol in your 20s, it actually is a linear process?
Well, we don't know that for sure.
Okay. I'll have to do this out of order then. All right, this is a question that I'm going to ask the audience to answer first. So think carefully. Aging affects a woman's brain differently than a man's. True? How many say true? And false? And Adam?
That's a loaded question. I would have to say true, just because of the influence of hormones on the brain that I'm sure everyone is aware of. You don't need a scientist standing in front of the room to tell you that, right? So menopause is associated with a lot of changes in cognition, and estrogen has a massive impact on the transmitters, the neurotransmitters in the brain that are involved in memory and attention and cognition. So they are different just because of different hormone influence.
I want to go back to the brain shrinking. Is it actually the neurons that shrink, or is it some other structure?
So at one point in time, we thought that the brain was shrinking because you were losing brain cells. And this is not true necessarily. So more advanced methods of counting brain cells, of course you can't count them all. So you need these complex mechanisms of sampling and doing a random sampling and then extrapolating out to the whole size, has shown us that the brain doesn't actually lose all that many cells with age. What happens is that the larger cells get less and the smaller cells become more. And what we now believe is that they're really just changing places. The cells themselves are shrinking. The connections between brain areas, the axons, are covered with myelin that's full of water, and that seems to decrease in size too. So it's more of a shrinking of the actual neural substrates rather than a loss of cells, which we look at from a pretty optimistic perspective, because if the substance is still there, it gives us something that we can work with to improve.
Okay, does stress impair memory, true or false?
True, definitely true. So stress and the hormones associated with like cortisol have a strong impact on the hippocampus, the part of your brain that's involved. There's two of them on each side of your brain that's involved in new memory formation. And there's a lot of research showing that it changes the dendritic, the branching of neurons and other chemicals required for memory.
Does that include the stress of being forgetful, making you stressed?
It might be a bit of a circle.
Okay, all right. All right, genes are more important than lifestyle when it comes to cognitive abilities, true or false?
You can never answer true to a question that has genes and environment in the same question. So it's always both. It's an easy one.
Okay. The brain starts to show signs of decline when we're in our 20s. We've already answered that one. The answer is yes. I'm also curious about this. The grooves in the surface of the brain widen with age. Is that true?
That's true. So the grooves, or the sulci and the gyri are the mountains, and that's where the cortex is. And as you get older, especially with things like Alzheimer's disease, but even normal aging, you have the shrinkage of the substance of the brain, so that makes the grooves, the sulci, become wider. Just to comment on the decreasing of cognitive abilities early on. So we used to think that aging was associated with cognitive loss in old age, but now we think of it as aging, starting at sort of the peak, literally being in the early 20s. And some things decline in a pretty straight, what we call linear way throughout life, like working memory, the ability to hold information in mind for very short periods of time and use it. The type of memory that if someone gave you a phone number, you would hold in your mind just until you got into the phone. So that declines from 20s on. Speed of processing, how quickly you take in information, how quickly you respond to things, same sort of pattern, even long-term memory. Some things are fairly stable, like vocabulary and semantic memory, so knowledge of the world around you. That could be very stable, even decrease later in life. And some types of cognition increase with life, like wisdom, taking your intelligence and your experiences and integrating them so that you have better decision-making ability. So it's not an entirely gloomy picture, but a lot of the things that we use daily do decrease right across life.
Okay, here's another one for the audience. We just have to assume that we're going to become more forgetful as we age. True? False?
I would say largely that's true. By far the universal signs are that if forgetfulness is decreasing short-term memory, which is probably what most people are thinking about when they think of forgetfulness, then that is something that decreases throughout life.
Okay, last question, for me anyway. The brain is so plastic that it actually changes as it learns. True or false?
That is true. So plasticity, again, another misconception about the brain is that it's static, and that is something how we used to think about the brain, that it was like concrete, solid, that once it formed, it stayed that way. That's not true. There's a property of the brain called plasticity, and plasticity means that the brain is constantly reforming itself, modulating itself. This is what occurs during all types of learning and happens throughout life.
So this is actually a structural change we're talking about?
Both structural, chemical, and functional changes.
Okay, you think their brains are ready to be plasticized?
I think they're ready.
Okay. It's all yours.
Okay.
So why don't we take a few questions before then? But quick question, are people hot?
Yes.
Okay, if you're by that front door, can you somehow just leave it open? Yeah, we're going to come to you in a second. We cannot open that door. Yes. I always worry that no one will ever show up to these. Well, when you can, open that door and try to leave it open if that's all possible. I'm going to go around and take a couple questions from the audience. And is there food piled up here that's waiting for people? Sandy? So if there's food piled up there, just yell it out so people can get their food.
Caroline.
Oh, there.
Peter.
Okay, so first question's right here.
What about those brain games that they're hawking on NPR for membership? Do they do anything to help improve the overall performance? Should you exercise your brain?
It's a great question. I'm going to hold that question because I have a slide on it at the end. The question is brain games that hopefully are meant to improve your memory. Do they really work? So I'm going to talk about that at the end.
I'm actually going to move in the back because these people are suffering back here. So who has a question back here in the middle? Great. Here we go.
As a musician, I feel that memory is incredibly important to performing music, and a lot of my colleagues, we've talked about this actually pretty recently. I'm wondering, to what extent, or have you observed or at all, if musicians tend to keep their capacity for memory longer simply because they're actively using it on a daily basis, anything like that? Performers in general. Whatever.
Yeah, I think
But before you answer that, Adam, so you're a musician. How old are you?
30.
So have you noticed your own memory around music has increased or decreased with age? Has it been affected at all?
I've actually felt that it's gotten better in the last 10 years. I've been a professional musician for about 15 years, and it seems to get better, so
there's a good amount of data that people that use their brains actively throughout their lives have higher, what we call cognitive reserve, and will have things like Alzheimer's disease, or at least dementia, present later. So that is true. There's certainly a lot to be said, probably more so than the brain exercises, although they do have a certain function. In general, is keeping your brain active by engaging and challenging of it.
So is it a net positive that I come to a science cafe and have 3 beers, or is that... question in the back. Okay, right here.
Why do I forget names, even if I remember faces so well?
Great question. That's a pretty common one for me. The name problem starts much earlier in age than most people realize. Probably most people in the room have had difficulty with that. My answer for that is that remembering a name is not an easy process. We take it for granted because it's something we do all the time and it's so important for our social interactions, but it's actually pretty complicated from a cognitive point of view because it's very abstract, right? The person's name doesn't really mean anything about them. It's sort of just assigned to them. It's not like when people's names used to be Farmer and, Goldsmith, and they actually were. So, it's an abstract association between a face and a name. And abstract associations are actually pretty hard to do. So if you're constantly doing it, there's going to be a lot of cases where you don't remember it. A lot of what occurs when you're trying to remember a name is that you're not listening anymore when the person is answering. Does anyone have that experience? So you are actually the one that asks for their name, and then you start doing something else in your mind. That's not a good way to remember things. We talk a lot about the interface of attention and memory.
We're going to take one last question and allow Adam to get into his slides.
Okay, there's been much made of the fact that new brain cells can be formed rather than just losing them over life. But how many of them do get formed? I mean, do the new brain cells really make up for the deficits of the dying ones?
Yeah, the question is about new brain cells. And again, another misconception about the brain was that there was no new brain cells formed after birth, and that's not true. There's something called neurogenesis that occurs in the adult brain. We don't know a lot about it yet. It's a fairly recent discovery. There are certain parts of the brain that it forms. In most parts of the brain, new cells do not form. So the hippocampus, the part of your brain responsible for new memory formation, is a part that has new brain cells. We don't know for sure if this somehow balances out the loss of structure and chemicals that occur with aging. But we do know some things. We know that exercise seems to increase the number of these cells in the brain through a nerve growth factor. So I'm going to talk about that at the end, but at least this seems to be a relationship between keeping yourself healthy through physical fitness and generating new brain cells.
It's slideshow time. It's time for the slides. Can I ask one thing? If you're in the table here in the middle, can you guys squeeze together just a little bit? Because there's a lot of guys in the back that are just in like mice back there. Squeeze your tables together a little bit. Get a little friendlier. Create a little more space. There's going to be plenty of time for questions during the presentation and after the break. from after the slide presentation, which is about 15, 20 minutes, we're going to have plenty of questions.
Okay, I'm going to begin. Sorry, it's crowded back there. So I'm going to talk about the aging brain and mind. It's really just going to be an extension of what we've already been talking about since we've been here today. I'm just going to talk about two things today. So this is my very short outline. I made this talk a lot shorter because I knew we were going to have a lot of questions. So the first, I'm going to give you an overview of research in my lab, tell you about the tools that we use and what our questions are. I'm going to even have you go through one of our experiments right now, sitting at your chair. And then I'm going to talk a little bit about what we now know about preventing or delaying cognitive aging, which is this term that we use for the change in the brain that's associated with cognitive loss, attention memory changes. These are some of my motivations for doing research on aging in the brain. I'll try to back up. The first is that the population, we didn't talk about this, but the population is aging. So it's, the population as a whole is getting older. The baby boomers have just reached 60 years of age. So there's a lot more people that are in this category where they're worried about their cognitive abilities.
Are you talking about the United States or the planet?
That's true throughout the planet. Well, throughout developed countries. It's not true for all countries, but it's true throughout. But it's very true in the US, obviously for the baby boomers. It's not just the age that's changing of the population, but the expectations are changing. This is something that I see all the time. So when I see patients at UCSF, they might be in their 60 years of age range, and they are very concerned about their cognitive abilities because most of them are still demanding a lot out of themselves. So I see a lot of people, for instance, this woman I saw recently is moving to Spain and she wants to learn Spanish. So we see a lot of people in their 60s and 70s that are learning second languages, and we see a lot of people that don't want to retire. They like the jobs, they work their whole lives to get in a position that they really enjoyed, and even though their workforce is full of 20-year-olds, they want to compete, they want to be on the exact same level. So not just the age is changing, but expectations place a big demand on maintaining cognitive abilities, not at an adequate level, but at an optimal level. Age-related cognitive declines do not just affect memory, but they affect a wide range of abilities. Attention, memory, visual spatial abilities, language, a lot of these things decline. And that even, and this sort of goes back to the point that I made, that even very mild cognitive deficits can decrease the quality of life, especially when people are trying to live very high quality lives in their older years and not just necessarily retire and watch TV all day, but really try to push themselves, even if they're not working. Then this decrease in cognition is really troubling to people. So what we do in our lab is we're trying to find deficits in underlying neural mechanisms. You might not be able to read these, but I think there's only slides with words on it, that underlie this broad spectrum of changes that we're seeing. Okay, so this is the general overview of our lab, which is located right over here at the Mission Bay campus. And the first thing we try to do is figure out how it works. And by it, I'm going to define a little better. But we use three different techniques: functional MRI, electroencephalography, and transcranial magnetic stimulation. I'm going to tell you about these two. I'm not going to talk that much about TMS. But what TMS is, it allows us to apply a magnetic pulse to the scalp, causes the neurons underneath to fire. So if you do it over the right motor cortex, the fingers on the left hand move. And we can use it to see in a causal way what brain areas do by stimulating or even suppressing brain areas. But I'm not going to talk more about that today. I'll tell you about these in a second. So once we define these mechanisms of how the brain works, we could ask the question, what goes wrong? How do these mechanisms change as people get older, either in the case of dementia or even in the case of normal aging? And then in our lab, we're also interested, now that we know what's happening, what can we do to fix it? So we're looking at two ways: cognitive training, sort of video games to train the older brain, and also pharmacology, like drugs that we use to treat diseases, although normal aging is not a disease. So I'll tell you about how we put all this together. Let me tell you about fMRI. So who's heard of fMRI here? That's a lot. All right, so fMRI is the most commonly used way to study the human brain in terms of cognition. It's only been around since the mid-90s, so it's not very old. It is essentially MRI. So the machine itself is the same as an MRI machine. What makes the difference is the physics that we use and the sequences to see what's occurring in the brain. Another thing that makes it different is how we use it. So as opposed to a normal MRI where you lay in the scanner and we look at the structure of your brain, in an fMRI experiment, you lay in the scanner and you actually do something. So you're lying there and you might be looking at a screen. We're presenting you with what amounts to like a very well-controlled video game. And you're doing experiments and you have a button box over here or a joystick. So you're responding to the stimuli and we're recording what's going on in your brain, but we're also recording your responses, your behavior, your performance, how well you're remembering things, how quickly you're forgetting, and other aspects. We can also record eye movements and other aspects of physiology, like respiratory rate. We can even put an EEG machine in there. These blobs that you see over here, we color them ourselves. They don't actually come colored. So what they are, these blobs represent significance. They're really statistical tests. And what happens is that blood flow changes in your brain in a very specific way when you're using a part of your brain. So for instance, if your eyes are closed and you image the brain and then you open your eyes, you'll see more activity in the visual part of your brain because there's more blood flow. So this is an indirect measure of activity in the brain. This is a little bit more. So in addition to looking at the functional activity in the brain, we get all this beautiful structure. Right now we could see down to a millimeter, which is really small, a cubic millimeter of brain structure. You could see that we could get very detailed views of the vasculature in the brain. This is not an injection. This is all just using MRI scans. And what this is functional connectivity. This is something we do in our lab. So this is not a map of brain activity. It's a map of brain connectivity. So you could define an area of the brain and see how other areas are communicating with it. And that's what's showing you right here. This is actually an experiment from our lab. And with this map are the areas that are connected with the visual part of the brain when you're thinking of a face. You could see that it's a part that just doesn't involve the visual parts, but involves other parts that communicate with it. What this is another type of connectivity map. It's called diffusion tensor imaging, DTI. And what this is a structural view of the axons, what I described to you before. These are the highways that connect different brain areas. So these are not vessels. These are axon pathways that we can now image that look at how brain areas are connected structurally. So a lot of what we do is take all of this information together from one single scan to give us a much more comprehensive view of what's occurring. So here is our fMRI center at UCSF. So we had set this up, and this is what we were talking about earlier.
Let me pause you for just a quick second.
Sure.
Does anyone have any questions about fMRI, how it works, limitations, anything like that? Oh.
How convenient.
You have a question?
So throughout all of these different things, what you're actually observing with the MRI, though, is oxygen flow, or the blood flow itself. So in the axons, you're actually looking at the blood flow and the oxygenation of it. And is it with isotopes, or how do you identify that stuff?
No, because it's looking at blood flow. So oxygen, whether it's deoxygenated or oxygenated, the iron part of it is exposed, has a different magnetic field. So basically it's the ratio of oxygen to deoxygenated blood. So we call it neural activity, but it's not. It's a blood flow measure of neural activity, and there are limitations because of that. It's a lot slower than real brain activity. Real brain activity takes place on the level of milliseconds, but blood flow takes place on the level of seconds. And that's a good segue to the next technique that we use, EEG.
We'll take one more question.
Yeah, I'm just wondering, I mean, I've had MRIs, and it was so noisy, I was going nuts. Are fMRIs that noisy, and if so, how does that not taint the results? Because it's so annoying and distracting.
Yeah, it is noisy. We have taken measures to decrease that. So we have special noise cancellation headphones. We have, we've worked on, we put a lot of time into trying to decrease that. Matter of fact, a lot of fMRI experiments are not visual experiments like the ones in my lab, but they're auditory experiments. So we can even present information through the headphones that you respond to. Okay, so here's our scanner. You can see it's set up over here. We have a very sort of nice hardwood floor and blue walls, very soothing environment, very Northern California for our participants. And it's important because these experiments are demanding, and the scanner is not necessarily the most comfortable environment to be in, and we want people to perform as well as they can. We also have another technique, EEG, and what this is a dense, there's 64 electrodes in this sort of swimmer cap that you put on your head. And if you could see, doing an EEG experiment makes you very happy, so I encourage anyone that wants to come down to the lab to participate. And what EEG does, unlike fMRI, is it's a much more direct measure of brain activity. So it's very fast. We could see when things are happening in the brain as opposed to where they are. The problem with EEG is we could see when they're happening, but we can't see where they're happening very well. So we tend to do both of these experiments. So if you participate in an experiment in our lab, one day you might do it under fMRI, and the other day you might do the same type of experiment with EEG. Okay, so that's enough technology. Now I'm going to tell you a little bit about what we actually study in the lab. So I'm very interested in this interface between attention and memory. So we all know from our experience, just of being alive, that there's an interaction between the two, right? So if you look at these two pictures, you'll see this woman reading a book in a quiet room, and you see this guy over here sitting at this table. Who do you think is more likely to walk away and the next day remember details about these events? Right, this is an easy one, right? So this woman sitting over here. And why this is because attention and memory interact. And this was really described nicely by Samuel Johnson in 1759. And he said that the true art of memory is really the art of attention. That no man will read with much advantage who is not able at pleasure to evacuate his mind. If the repositories of thought are already full, what can they receive? If the mind is employed on the past or future, the book will be held before the eyes in vain. So it was really nicely described. It took me a long time to actually find a quote that describes this, but I think all good talk should have a nice quote in it. But a lot of people have not really commented on that because it took me a long time to find that. But I think it really speaks exactly to what I'm trying to study. And now I'm going to define my perspective and my terms of thinking about this. So let's start with perception. Right? Simply defined, perception is how you view the world around you. We now know that perception is not a passive process, so the world just doesn't flood into your brain. It's influenced by attention, and there are really two main types of attention that can be thought to influence perception. One is external, or stimulus-driven attention. So this is happening all around us, right? So if there's a flash of light or a loud sound, or even if someone calls your name quietly behind you, you pay attention whether or not those were your goals, right? So you might be battling right now, hearing orders being called out, or in a restaurant when you're trying to have a conversation. So this is the environment sort of imposing itself upon you and how you perceive the world. The other type of attention is what most of you are doing now, is goal-directed attention, right? So you're choosing to pay attention to what I'm saying, to looking at the slides, and thus that's commanding what you perceive. Every one of your interactions in the world, in your environment, involve a balance between these two types of influences. One, your goal-directed control over how you perceive the world, and the other, the environment imposing itself upon you. I'm interested in how these two types of attention, another way of looking at this is bottom-up processing and top-down modulation. And I'll tell you what I mean by modulation in a second. And how this goes out and influences both working memory, short-term memory, and long-term memory. So let me try to describe this in a more intuitive way. So take a look at this scene over here. We have this gentleman that appears to be involved in this table, right? He's sitting at a table with three other people and should be having a conversation with them, but he's not, right? Because there's a strong bottom-up influence, some sort of conversation here that's going on that must be very important because it's actually drawn the attention of several people throughout the room. But despite the fact that he's failing his top-down goals of being involved in this conversation because of this bottom-up input, it's not universally overwhelming, right? We have a woman over here who's so engaged in her text message that even though she's the closest to this, she's able to adequately ignore it. So we see this all the time, this balance of attention and ignoring. And what it tells us is that there's really two faces of top-down modulation. There's both focus, and the flip side is that there's ignoring. What we're starting to realize from our research, and I'm going to give you some examples, is that it's not two sides of the same coin. That focus and ignore are actually separate processes, and one could be working and the other could be failing, so they likely have different mechanisms that control them. I'm going to tell you about that. Let me just show you what happens in the brain when this occurs. So this is the brain, and this is the front of the brain. The eyes would be over here. And interesting enough, when you look at something, the first part of your brain, the cortex that processes it, is the occipital cortex in the back. So you can see it activates over there. And the information then travels forward. Actually, there are two streams. Travels forward, and what happens are that the attributes of the stimuli around you get integrated together. And you have these more complex representations of the world as you come forward. This is another way of looking at bottom-up processing, just how you process the world. We think that there are then connections from the prefrontal cortex, the very front of the brain, that project down and modulate, this is why we call it modulation, modulate the activity based on your goals. More activity for when something is relevant, less activity when something's irrelevant. Again, you can see these two forces interacting. The prefrontal cortex is an interesting part of the brain that I'm just going to pause on and tell you the story of Phineas Gage. So who here knows the story of Phineas Gage? Wow, that's Pretty amazing. They say he's the most famous neurological patient, and I can see that's probably true. So Phineas Gage was a patient, or had this unfortunate incident occur in September of 1948. And he's a 25-year-old. He was a railroad foreman in Vermont. And Phineas had the job of pouring the gunpowder into the holes when they were dynamiting out land to make rooms for the railroad tracks. And they had to put some gunpowder in, and then put some sand in, and then tamp it down. And apparently he was distracted, which is sort of ironic given the rest of the talk, and he tamped it down before he put the sand in, and it exploded and drove this tremendous tamping iron, 13-pound tamping iron, through under his left eye socket, up through his frontal cortex of his brain, where it landed 60 feet behind him. And if that's amazing, it's probably not the most amazing part. The most amazing part is that Phineas Gage lived, and he walked, or he was taken, he actually walked out of that scene, was taken to a local hospital, and he survived this. Interestingly enough, everyone thought that the frontal part of the brain must do nothing because Phineas seemed fine, right? But after further reflection, it realized that Phineas was no longer Gage. So I'm just going to quickly read you some notes from his doctor upon evaluating him. By the way, this is a sort of shocking picture to show you how large this tamping iron was next to Phineas's head over there. So he was fitful, irreverent, indulging at times in the grossest profanity, impatient of restraint or advice when in conflict with his desires, obstinate, devising many plans of operation, which were no sooner arranged than they are abandoned in turn of others more feasible. The balance between his intellectual faculties and animal propensities seemed to have been destroyed. So we see here, this is a beautiful description of someone with frontal lobe damage. And what happens is they have something called disinhibition. They've lost control over how they regulate their behavior. And we know that it's not just control of behavior, as I'm going to show you, it's also control of how you perceive the world. So top-down modulation is a control process. You control how you perceive the world, and you control how you act in the world. And you can see that last description was really brilliant because the prefrontal cortex is the part of our brain that has evolved the most, largely in its connections with the rest of the brain. You could see the percentage of the brain that is dominated by the prefrontal cortex increases throughout evolution all the way up until humans. Okay, so I'm now going to tell you about an experiment in our lab to look at top-down modulation, how it changes with aging. Any general question before we move on? All right.
Any questions? No? Oh, there's a question right there. All right, you're going to have to repeat that.
I'll repeat it. The question was, how do you know Phineas wasn't like that? That's a good question. From what I told you, that might be the case, but that's not the case. That Phineas was actually this very church-going, generally likable and well-controlled guy before that. So it was the change in his behavior which was so shocking. All right, so I'm going to tell you about this experiment on top-down modulation with aging. So I'm going to tell you about one experiment that we've published in several papers over the years. And this was done with both fMRI and EEG in a group of healthy, younger, and older participants. This was done when I was at Berkeley. The age range of the younger folks were 18 to 30, and the older participants were 60 to 80. And they were very healthy. It actually took us a long time to find the population quite this healthy. So they had no medical conditions, they had no depression, on no meds. They were not even concerned about their own cognitive abilities. They felt that they were doing pretty well. So we found this population, and I'm going to tell you about the results now. So before I start, I'm going to have you perform one trial from this experiment. So there's many different trials. I'm going to tell you what they are, but you're going to do one of them now. So what you're going to see are 4 pictures. 2 pictures of faces, and 2 pictures of landscapes, of scenes. And your goal is to remember the faces and ignore the scenes, because the scenes are not relevant for this task. I'm just going to test you on the face. So you're going to remember those faces. It's going to be an X in the middle of the screen, and then 9 seconds are going to pass. And during that time, you're going to remember those faces, and then I'm going to show you a face. And if you were doing this experiment in the scanner, you'd press the right button for yes, that was one of the 2 faces, and the left button for no, it wasn't. So for now, you just have to think yes or no. Was that the face, or was it not? Okay, here we go.
You can hear the murmur of the audience saying it out loud.
All right, we get a second chance. Second chance. I don't know why it didn't show it.
You did show it.
Did it? Was I just not looking?
You were exactly.
I was taking a little break. All right, so is that the face? Yes or no? No. It's not the face. I should ask you the first time, but some people got it. So if you thought that was the face, it's okay, because most people do not get this correct the first time, and there's a whole practice. So what we can now do is we could look at the experiment in different trials. So here's the one you did. Remember the face, ignore the scenes, hold it in mind, is that the face? And then there's the remember the scenes, ignore the face trial, which is the exact opposite. So it would look just the same, but now the scenes are relevant. And then there's a passive view trial, where you just look at the faces and the scenes, you let them flood over you, you have nothing to remember, you just press the button here for the direction of an arrow. So what we have is that during this condition over here, all the stimuli are balanced. So there's the same bottom-up information, the same information coming into you from the environment. So any difference in brain activity between these three trials must be due to top-down modulation, right? If you were, let's say, anesthetized and your eyes were held open and you were shown these three trials, your brain activity would look exactly the same because you're seeing the same amount of stuff. That's what the passive view does. Not quite as perfectly as anesthetizing people, which we don't really do in our experiments, although it would be good. But that's supposed to show you what happens when you're not trying to remember or ignore. So now what we can do is we could look in an area of the brain that is specialized for detecting scenes. There's another area specialized for face, believe it or not, and this has already been well described. So we just use this area, let's say the scene area. I'm just going to tell you about half of the experiment. The same thing happens on the flip side. If we look in the scene area, we can compare how activity is different between when you're remembering scenes, passively viewing scenes, or ignoring scenes, and see if there's a change in activity. So when we look at our young subjects, what we find is that there's this big, robust response, and this is an average across 20 people. When they're ignoring scenes, this is the trial that you did, paying attention to the face, ignoring scenes, there's much less activity. And when you're passively viewing scenes, it splits right in the middle. So what this offers us is a measure of enhancement. How much more activity you have when you're paying attention to scenes as passively viewing, and suppression. How much less activity you have when you're ignoring versus passively viewing. Okay, does that make sense? So we want to look at these two measures and then look what happens when the older subjects do exactly the same experiment. So we have here enhancement and suppression. Does anyone know this finding? Okay.
Not somebody that's in his lab. Well, let's take a guess, actually. What do people think that's going to look like? Just yell it out.
They all ignore.
They're saying it's good. Most people are saying it's going to be close. It's not going to be that dramatically different.
Okay, well, that's not the answer, because else I probably wouldn't be standing here today talking to you. This was actually a very exciting finding. The answer is pretty dramatic, but not quite in necessarily the way you might guess. So enhancement is preserved. They look like they're 20 years old if you look at how much more activity there is for relevant versus passive. The big difference comes in the irrelevant information. So they're not suppressing relative to passive view. There is still less than remember, but this difference is not significant. You can see it's pretty dramatic compared to the younger subjects. So those scenes that were coming up that were irrelevant, in general, by looking at brain activity, there's no other way of really knowing this, because remember, the response is just to the faces. We can see what's happening in the brain when the scenes are being presented and see that there's more attention being directed to it than the younger subjects, right? But that's only part of the story, right? The other part is that it's a memory experiment, and they don't remember the faces as well, the older subjects, as the younger subjects. And although they did fairly well, 80%, it was significantly less than the 90% of the younger population. The interesting part is that when you split the population and you look at the older subjects, this is their accuracy scores. Here's the high-performing older adults. So statistically, these were no different than the 20-year-olds. Then you have a population over here that were more than 2 standard deviations away, so significantly impaired on this very simple memory task. When you look at the suppression levels in their brain for ignoring information, it's only the lower performing older adults on remembering the faces that were unable to significantly suppress the scenes. So what we here have is a direct relationship between attention and memory, but not in the classic way of focus as most people think of attention. Rather, how well you ignore information directly relates to how well you remember things that are relevant. And although I'm not going to show you, there's a direct correlation between the degree of suppression and how well they're remembered. Another interesting thing is, so when people finish our experiments, they're sort of relieved because it's been a long time, they've been inside the scanner, and then we surprise them with another experiment. This is very endearing to them. And we show them all of the pictures that they saw during the experiment. And we also show them a whole new set, an equal number of new pictures. And they have to press a button from 1 to 4, whether or not they think that they saw this picture or it's a new one. And most people think that we're wasting our time because they're not really sure if they saw the picture. But it's definitely not waste of time because almost every subject remembers, although even if they're not exactly sure, the pictures that they saw versus the pictures that they were ignoring. The really interesting thing are these scenes that were supposed to have been ignored, right? So who remembers the scenes that I showed you before? Right? You shouldn't remember them, right? You're supposed to be remembering the faces. What we find is that the lower performing older adults, the ones that did not do as well on this test, remembered these scenes better than the higher performing older adults, and they actually remembered them better than the younger subjects. So actually they had better memory for things they should not have been remembering. So really showing you that it's not quite as simple as saying that memory changes with age. We really see that their memory is actually fairly good for some things, right? But it's really the interaction of how they're balancing what's relevant and irrelevant. Just to tell you a paper that just came out a couple of months ago, a second in the brain is a very long time, right? A lot of processing occurs. And we were curious, when in the brain does this suppression deficit occur in older adults? So we did the same experiment with EEG, that, as I said, allows you to look at the timing of information. And we found, I'm not going to go into the details here, but we found all of these measures in the first 0.2 of a second, early measures, and then we found measures later in the second, right? So we could break up the second. And what we found is that the older subjects only had a suppression deficit in the very earliest measures. By the end of the second, they looked like they were young adults. So this leads us to believe that it's not that old adults have entirely lost the ability to suppress, it's that they're slower at it. And by that time, it's too late. Right? So if you let too much irrelevant information in, it causes interference because you only have a limited capacity. So if you let things that are not relevant to your goals, they cause interference and they decrease your ability to remember. And even if you could suppress it later on, it's suppressing it quickly enough that's really important. And what this is another common way of looking at the brain. It's called the processing speed hypothesis. And it's that a lot of the abilities of the older brain, the deficits, are really defined by a decrease in the speed of processing. And you can see that it's occurring here too, but it's really selective for ignoring more so than paying attention.
Let's take a couple questions.
That's it. That's the summary for this part of the talk.
Oh, do you want to just go on by the summary while I try to
this is
get
when
to somebody
I
with
was
a
going to
question?
pause now. I'll just tell you the 3 points, and then you can ask questions about this. So normal aging is associated with a selective deficit of top-down suppression. Suppression is not abolished with aging, but it is just too slow. And the results establish this critical relationship between attention and memory impairments in aging.
Okay, first question is Ben.
Okay. I guess the question is, was this inability to suppress irrespective of whether it was a face or a landscape?
Yeah,
So it goes all
so
across.
the flip side of this is that the same things occur for the faces. So I was just, in the sake of time, I was trying to not show you all the data, but it's the same exact thing.
Are there other modes besides faces and
Yeah.
landscapes that
Yes.
you've noticed in the brain?
So we just did this exact same experiment with dots that were different colors or they were moving. And you have to remember the color or you have to remember the motion of dot, and you can see the same type of effects. Actually, because someone might ask this question, so I'll just preempt you. We did not find this relationship between ignoring and remembering in the younger adults, largely because they really did too well and we didn't have enough variability. But in this new version of the experiment, which we just submitted for publication, it's a lot harder to remember the colors and the motion. And now we can see that there's a very nice relationship, even across the trials, that on those trials that you do not do as well, you were letting in too much of the irrelevant information, and these were 20-year-olds. So it's not just an aging phenomenon, it's true for everyone.
I have a question. So this sounds kind of weird, but since we know that older people have the ability to remember irrelevant information, can we use that and harness that as to keep them engaged in society? I mean, the way that we know how we remember things, is there a way that activities or things that can keep them engaged in society?
So what are your ideas and thoughts behind that? How can you garner irrelevant information for a positive good?
I'm not quite sure. I'm open to learning.
So that slant on this type of data has already been espoused, and there was an article in the New York Times not so long ago where a researcher was making a claim that this is actually a effective part of aging, because older adults take in all this information. Like, you have to remember, in this experiment we tell you what's relevant and irrelevant. In real world, you don't always know that. And the claim was made that by taking in more, this is what contributes to increasing wisdom with age, that you have just a larger, broader experience. I don't think that's entirely true. I think that sometimes it's true when you don't know, but there's a lot of times that you do know what's relevant and irrelevant. And when you do know, it's important to really make a big contrast between them in your brain. But, you know, I get the point.
I'm going to keep making my way through. So if you're in the front or the back and you have a question, just keep your hand up, and I will remember them, and I will ignore the other hands.
Okay, so funny you should have chosen the landscapes and the faces. And what about somebody like me who has like a moderate level of prosopagnosia and topographical agnosia? What can you tell us?
Wait, you're going to have to tell us what that is.
Prosopagnosia, the difficulty learning people's faces, remembering their faces. Topographical agnosia, difficulty remembering what you've seen, landscapes, buildings, places.
Is this a diagnosis?
Yeah.
That's interesting. We should talk after this. So I think that, you know, yeah, exactly. If you want to participate in a study, we'd love to have you. Most people don't have those you know, changes in how they perceive, although it's a lot larger than I think was normally appreciated. I could say, you know, at least for the subjects in here, it wasn't that they were not able to remember the relevant information. It was just largely how they were letting in too much of the irrelevant. So I think if someone that had prosopagnosia was performing this experiment, they would do really, really horribly on it. Probably be below the level of a lot of these people. And that's why we are moving on to experiments on the dots and moving things to try to look at across a large range of stimuli.
I've read several articles recently about how the internet and our ability to multitask online is affecting the brain. And I was just wondering if there is anybody studying older adults who use the internet frequently and how that's affecting the way they age, or if you expect that that will be something that is studied in the future.
What did the article say?
Well, the Atlantic Monthly wrote one last month, I think, that the headline was, "Is Google Making Us Stupid?" And I think that the article basically said that it was affecting our ability to pay attention and our ability to retain information, because we're always hopping around and multitasking and not, like, that that we're not going deep enough, that we're doing a lot of things that we're skimming, and that the internet enables us to do that. And I can't exactly remember. I can't remember everything else about it, but it was really good. You should read it.
Yeah. So.
Well, do other people feel that way about the internet? It kind of makes them stupid. I feel that way about Facebook.
So it's probably safest to answer that on both sides, because I think there's an argument for both ways. I often say that I think that the increasing amount of technology in the world leads to these type of suppression problems. If there was a time when you didn't have a cell phone with texts and emails coming in all the time and computers with pop-up windows, and I think the amount of information that we're encounter in a per moment basis has increased throughout time. And that might be some of the changes that we see. Remember, this is a cross-sectional study, and a lot of these older adults did not grow up with the same type of need to suppress as much information. At the same point, there was an article published recently that talks about older adults that use the internet and the web have decreased cognitive deficits. Actually, it seems to help them by allowing them to engage. So I think there's a flip side to that as well.